actually quite surprised to see how much of interest there is still. Because part of this talk is probably published already over several social media. But anyway, I'd like to give an overview of everything that I have been working on in terms of biophysics. Not only biophotonics, I will show you other physics tools than photonics tools as well. Although I'm chiefly interested in the nano-optical research in our group, and you're welcome to visit our lab whenever you like. It's in the entrance floor of the department. I'm associated with also a micro and nanotechnology program, so we have in our group both graduate students from both physics and micro and nanotechnology department program as well. But we do collaborate with biologists and molecular biologists a lot. And in this talk, I will concentrate mainly on those collaborative work that we have been doing using tools available in physics, namely derived from micro and nanotechnological applications. Okay, so this is kind of a crossover technology from physics into biological sciences. What with very simple means one could contribute in understanding of biology at the molecular and cellular levels. Okay, so please try to see and look for problems in the broad sense, not only in solid or liquid or plasma. The world is beyond that. There is also soft matter, in which biological specimens are in this family. Now, you will see I prepared today 4 different examples. So there will be 4 parts to this talk, but all of them are centered around neurodegenerative diseases, fighting against neurodegenerative diseases, because it turns out to be a major problem in human biology, not only human, also animal. And I listed only a few of the well-known, most known versions of these neurodegenerative diseases. What's common to all of these neurodegenerative diseases are that they are actually protein diseases. They have to do with wrong kind of folding of otherwise functional proteins. When they fold in the wrong fashion, they become unfunctional or dysfunctional, and sometimes even toxic. Okay? There is always a question: what causes this misfolding? Is it misfolding? Can it be modeled like nucleation and growth, as in solid-state physics, or is it something more complicated? It turns out that there are no simple answers in biology. Okay. Okay, so let's go ahead quickly. In my first example, I will mention a group of proteins which cause the Prion disease, namely prion protein. Prions, are you familiar with Prion disease? Did you ever hear what prions? Maybe in Turkish.
Mad cow.
Exactly. In the flat language, you name it as mad cow disease. Okay, deli dana. Okay, so prion disease is one of the many like diseases in that family. There is not only mad cow, but also similar other diseases which are in this line. But prion disease is interesting because it's a contagious disease which is spread by proteins, not even viruses or bacteria. It's simply over proteins. You digest a mad cow meat, and then you can get mad cow disease. But it's not so bad, don't worry. Anyway, was a major problem back, I don't know, 15, 20 years ago. There was quite a big pandemic. It wasn't COVID back then, it was Prion, and people had suffered quite a lot. There was a certain interest in fighting Prion disease. And it turns out that this sort of proteins, like many other proteins, functional proteins in the cell, has this beta structure which forms so-called beta sheets. Beta sheets are segments of the protein which are appear in the form of layers, layer by layer folded proteins. And there are also some loose groups which allow for flexibility, etc. And there are also unstructured groups, alpha groups, etc. So helical groups you see here, then the beta groups is flat laminar. Now, and there's also completely unstructured kind of amorphous end to the protein. All of these different segments have a certain function in the folding and function in the cellular level. Now, if some modification somehow occurs in the protein and which causes a major difference in its folding through its sequence, it may cause some different form of folding which becomes less, let's say, soluble in liquid, in water, in aqueous solution. Then it simply precipitates. That's what chemistry says, right? So proteins need to be somewhat soluble in cytoplasm to have function. If they precipitate, they don't function, right? So it may simply, again, I'm oversimplifying the actual mechanism, but that's the major, let's say, dynamics behind why you get ill if your proteins start to misfold, okay? In the simplest one. So prion protein is also one of such example. And now when we take a look at precipitates of the prion protein, we see that it tends to form long so-called fibrils, okay, chain-like structures. And what you are looking at right now are atomic force microscope images of these proteins dispersed on flat substrates, especially designed very flat substrates, such that even the nanoscale topography of the folded protein can be highlighted. So we're using here the atomic force microscope, which is designed to generate topographical maps of nanoscale material. But we are looking at a soft matter, which is protein. Protein is something like a polymer. It's a biological polymer. You can call it that. Polymer is not like a planar two-dimensional one. It tries to make fibrils. Okay. There are reasons to that. Well, now what people have come up with to distinguish if the proteins are precipitating in the form of fibrils inside the cells or not, or they stay loose and functional, is a fluorescence assay. Its assay is a kind of methodology, let's say, methodology. It's fluorescence based. So it's simply like if you add a certain molecule, in this case a thioflavin T molecule, into the cytoplasm. Now, if there are fibrils forming, so the proteins is misfolding and causing fibrils and precipitating, then this molecule goes in between these beta sheets, and when it goes, it becomes fluorescent. So if you see fluorescence, you know in a kind of indirect fashion that you are forming fibrils. Okay. If it doesn't go in between these fibril beta sheets, and excess amount of beta sheets, then it doesn't fluoresce. Now what you can do is, you can check the time dependence of the onset of fluorescence how it fades, if it maintains itself, etc. You can use this, the fluorescence microscopy tool in physics, to just monitor the molecular dynamics in a cellular level. So, I mentioned so far atomic force microscopy and then fluorescence spectroscopy or microscopy, right? So, two tools already covered. Okay, now further with the atomic force microscopy, now you can associate different dynamics of this THD fluorescence curves with different outcomes of protein precipitation. Take a look. When you see fibrils forming or oligomers forming, so these are just simple fragments. They don't mean that there is fibrillization. But the difference between the morphology can be by use of AFM as an analytical tool, can be statistically studied in very detail. Okay? So if you make topographical features into a statistical distribution curve, you can see that there are certain distinct heights along the fibrils or oligomers, which correspond to probably single monomers of the otherwise polymeric substance. Okay. Now, having a careful analysis of this, you can discern single step sizes, differences between the heights. And, okay, if you had like a graphene sheet, everyone talks about graphene or 2D materials. If you have 1 layer, 2 layers, 3 layers, you could see the steps right at the borders easily. No way, no problem. But this is a highly irregular substance, right? It's derived directly from cells. So when you lay it on the surface, they don't make very structured two-dimensional materials, very regular, so you can see clearly the steps, etc. But what you could do is you could still compose a histogram, distribution of heights, as a function of frequency. With which frequency you observe a certain height. Okay? Then using the statistics, you can still discern what is the height of a single layer, what is the height of a double layer, etc., etc. Okay. So this is one sort of interesting work we did. I will not go into the details why we did it, what we learned, don't worry. This is only aimed to show you what people can do with the physical tools. Okay. Another example from the molecular system is the case of alpha-synuclein. This is another protein. This is naturally expressed protein in neurons. And if it misfolds and precipitates again, it causes a disease. And what people are trying to do is to interfere this precipitation by some drugs. How you can stop the molecule from precipitating to stay functional in the cytoplasm, etc. What disease does it cause? Any idea? Okay, I'll go back. Parkinson's. Okay. Parkinson's disease. Each of these diseases is associated with a protein. Okay. Alzheimer's is misfolding of beta protein. Parkinson's is misfolding of alpha-synuclein. Anyway, there are several other diseases. Even diabetes is a protein disease. It's misfolding of insulin, becoming insoluble, dysfunctional, cannot regulate your blood sugar level. Okay. So, doctors wish every disease is simple to fight. However, every protein can misfold and cause a different disease. Life is not so simple to maintain. Now, what one can do in order to look for drugs, what would you do first? There are zillion different molecules you can think of. Can you test each of them one by one? if for potential drug as a potential drug? No, there is no way that one can do such a test in the brute force fashion, right? Why? Because number of molecules is exceedingly high, beyond imagination. So, in that case, what we do is, we first start with what biology does, the cell does. The cell operates based on certain hormones, and hormones are what? Are just other proteins. Okay, so in that sense, dopamine is found to regulate the alpha-synuclein fibrillation mechanism. Okay. It is found naturally because dopamine is everywhere in the brain. Okay. All the neurons express dopamine and many other hormones likewise. Now, what we started to look for are analogs to dopamine which could mimic its action. Okay? Now, how to look for it? You can first make a detailed structure of dopamine using nuclear magnetic resonance, NMR, and then try to figure out certain candidate molecules out of at least a list of 100 molecules maybe. Now you can reduce the zillion molecules to 100, which are known to be analogous to dopamine. Okay? This is the first level. Second level, not all 100 of them will do the job. You have to first reduce the set further. How can you do that? You can, for example, run some simulations, molecular docking simulations. If the molecules don't like to dock onto themselves, other proteins, other molecules, they simply don't have chance to interact. Okay, that's the simplistic approach. So what you do first, you try to make a simulation of molecular docking on protein, alpha-synuclein. Okay, this is what people have done. And we reduced the 100 analogs, the list of 100, to about 10. 10 candidate molecules. And out of 10, we had to select yet 5 of them for practical reasons. Still, 10 can be quite extensive, exhaustive to test in the lab. But for this study, we just picked 5 and we went ahead. We checked by introducing these molecules if we could have ended up with the same effect as the dopamine. So you see, alpha-synuclein protein generates again fibrils, pretty much like the prion protein. But when you add the dopamine, you only get oligomers. The fibrils don't form. Okay. Now, this is the control experiment. This is the experiment with different candidate molecules. Okay. So this one has no drug molecule, only alpha-synuclein. You see long chains of fibrils. Definitely has something wrong with it. It shouldn't have precipitated, right? Okay, now we check by addition of these candidates if we can alter the situation. Again, AFM is helping us to discover that. So we see that they do, some of them have an effect to a larger extent, some of them not much, you see. So it turns out that even if we were able to reduce the set of 100 candidates to 10, and then select 5 most likely out of them, and we do the experiment, we see that actually none of them works as well as dopamine, but some may approach somewhat, like in this case. Even this is partially, there are long fibrils around, a bit of its action. So again, not every study ends up in great success in discovering drugs. Okay? That's the truth. So here I show you yet another set of images. This time acquired using another tool, transmission electron microscope. So the third tool. Now in here, you literally see with your eyes, atom by atom, the fibrils, and you can check how small these polymer pieces, fragments, segments are in more detail. Okay. So, at the end of the study, what you can conclude? Well, I showed you high-resolution microscopy techniques of atomic force microscopy and transmission electron microscopy. I mentioned about NMR, etc. So, especially high-resolution microscopy techniques are helpful to provide direct evidences to your results, but they can be combined by other techniques like fluorescence, etc., which can provide indirect also indications. So you have to use a multi-dimensional approach to actually figure out what's going on in a highly complex problem, such as this biological problem. Okay, so, and then using a combinatorial approach of computational biology and high-resolution microscopy, you can, if you can't let's say discover, you can't say we discovered, but you can at least screen potential drugs. Okay, so good. Now, other two examples will come from cell models. So not molecular level, but cellular level studies. Here, this example is from infrared spectroscopy with microscale resolution, again around prion infection. Okay, so as I said back then, we had a strong impetus to work fight against prion disease. So this is one other study as such. Now, in order to do highly sensitive infrared spectroscopic measurements, you need strong and collimated infrared light sources. Okay, and they're unfortunately not that widely available. In our case, we were lucky because we had a synchrotron radiation back in Italy, in Elettra. There was an Electron Accelerator. Now, you can generate every photon by Bremsstrahlung, by slowing down the charges, okay? Slowing down the charges. You first accelerate them, then when you break them, slow them down, they emit radiation. And the amount of emission energy depends on the, not the velocity, but the deceleration. Okay? So deceleration level decides what energetic photons you radiate. And if you are very gentle, you decelerate very, very slowly, you can generate quite a lot of infrared radiation, and moreover, over a very broad range, because you can slow down electrons at whichever deceleration you like, okay, using magnetic forces. All right, so, but of course there was a challenge. You would like to do some absorption measurements in order to figure out the chemical composition of the cell by doing infrared spectroscopy. But then you need a substrate which shouldn't absorb your light. Because if the substrate absorbs, then you don't get any signal from your cells. So we figured out that a very thin layer of silicon nitride does the job. It has a very flat, not zero, but flat absorption in infrared, which doesn't have features that you could mix with, confuse yourself with the signature of the cells. Okay. Okay, so here is what an infrared absorption spectrum looks like. In the absorption dimension, typically this is displayed backwards, inverse fashion in transmission. Maybe you are more familiar with that. But anyway, in the absorption spectrum, we see distinct peaks, energies, photon energies, which correspond to absorption of different constituents of the cell. What are the constituents of the cell? Well, we do have a cell membrane, which is a lipid bilayer, right? But there are also phospholipids. So some of them are phospholipids, lipids. Then you have quite a lot of proteins, both in the cytoplasm and the membrane. Then you have the nucleic acids in the nucleus, right? All right, so now what we did is we compared the chemical composition of prion-infected neurons versus normal neurons, uninfected neurons, to see if there was a spectral difference between the two. Okay. And at first glance, it seems unfortunately no, right? Looks like identical, but no. One has to do a more careful analysis, and one can do it by actually taking the difference between the two spectra, or actually you can make a spectrum more sensitive to changes by differentiating the difference. You first take a difference, then differentiate it. Differentiation highlights the slopes, right? So it gives you even a better kind of idea what differences take place. Okay, so with that, we will also, in order to associate absorption with absorbance, these are two different physical quantities, you have to know what volume of specimen absorbs your light. Okay, otherwise, absolute absorption is meaningless. However, absorbance of a material is meaningful. Okay, so absorption per distance traveled by light is absorbance, right? So in order to convert everything to absolute dimensions, you need to know how much of cell, how much of material does the absorption. How do we calculate the volume of a cell? Atomic force microscopy is showing us topography. So once I have the topography, I can calculate the volume of the cell. So what you do is, you do spectral and atomic force analysis on the same specimen. You end up with volume. Using the volume, you can convert absorption into absorbance. Then using that, you can take a look at the changes in the absorbance. This is dA over d lambda or something like that, d Wayne number. Now this is what I'm talking about. So it's the differential of the differences. And only in careful analysis of these, you see where the small differences lie, how slightly, you know. Why? Because it's really, really small amount of material. Okay, really small amount of material that does the difference. And now, once you figure out about these differences by this very careful analysis, you can come to some conclusions, such that if you compare the normal cell content to a prion-infected version, the light gray is the prion-infected, and the dark gray is the normal. If you see a statistically significant change between the content of proteins, lipids, phospholipids, and other proteins kinds, other bands, you can have an idea. Again, somewhat statistical, but it's a statistical analysis of spectral, detailed spectral study. Okay, which can be conclusive, and it turns out that there is a drop in the total protein content. Now, this is a bit of information we just learned from all this amount of work. The rest is, again, we still have a lot to do about solving the mysteries, but biologists and physicists have to work further on the problem. Okay, And these are some distribution of these different proteins and lipids and other components in the cell. Okay, you can make maps out of the distributions of these materials on a single cell level. Okay. All right. I'll go quickly through that. So it turns out that, again, by the way, this was fluorescence microscopy of the same sort of cells with different labels, different spectroscopic, fluorescent behavior, which also showed some statistical differences. This, again, yet another kind of technique, fluorescence microscopy, which we used on top of infrared absorption atomic force microscopy. So 3 techniques at least mentioned. So, in conclusion, infrared microspectroscopy provided detailed data on multiple molecular compositional channels in the cell. So it turns out that by just careful analysis of the spectra, you can discern if the cells are infected or not. It can be used as a diagnostic tool. I don't recommend. You can't go to a synchrotron each time you would like to do diagnosis, right? But in principle, it is evident that it can be used, right? So, but complementary techniques like fluorescence microscopy and atomic force microscopy, adding up to the infrared spectroscopy, helps interpret our results with higher reliability. The last, but not the least, example comes from our collaboration with Çağdaş Devrim Son of Molecular Biology, Biological Sciences Department. And it is, Şeyda is doing her master's thesis also on this subject. So we studied the behavior of neuron cells, again neurons, everything is around neurons. Their behavior on femtosecond laser micromachine silicon substrates. Well, I actually gave this presentation before, but just to give you the complete picture what physics tools can do in biological analysis, it's not only analysis. Okay, you can use physics tools also to modify material, to direct cells, to induce effects also. So far, I gave examples in only analysis part. Okay, this example is from modification and driving of the cell behavior towards certain directions. Okay, so we interfere now with the cell behavior. We are not just passive observers in like spectroscopists or AFM microscopists. Okay, so in this, the motivation was that if we can understand what sort of topologies can be used in modification of the behavior, such as the adhesion, proliferation, its shape, differentiation, and migration behavior, they can, only the topography under a cell could be used as a tool to drive the cells to take some certain actions which they don't normally do. Okay? It's a very simple approach, involves no chemistry. Okay? So the cells are in their native state, but what they walk on is engineered. Okay? So that's it. That's the beauty of simplistic approach of this tool, but it turns out that you can do quite a lot of things. So there are examples in literature. So, for example, you can use the topography to align them, which is something we have studied also here extensively. So, why neurons? Why? Because it's widely studied on different materials, and neurons are especially one of those cell lines which are susceptible to changes in the topography. Okay. Some epidermis cells could be as well, some muscle cells and bone cells are also, but neurons are quite receptive. And one futuristic idea could be if we could engineer a surface such that when we simply plant the cells, would they, in a self-organized fashion, form a neural network? If they do, then we are in business. We can make some computation even using them. Okay, this might be one of the drives behind this kind of work. All right, so, and we did the engineering of the surface by using femtosecond lasers, highly advanced, ultra-fast light sources, and we also used as a material silicon. Why? Two main reasons, don't worry the rest. Silicon is biocompatible, cells don't feel bad on them. And second reason, we know how to modify silicon with femtosecond lasers. Okay, essentially the two. All right, then, so we do have some initialization of the work, some structure, so substrate preparation like cleaning, etc. You start your cell culture, then seed them, grow on the substrates. Then you have to inspect their motion, their layout using microscopy. It could be SEM, scanning electron microscopy. It could be fluorescence microscopy. And two other tools. And here is the laser structuring effect. So what you see is from self-organized patterns forming on surface of a solid material, such as a metal or semiconductor surface, which is simply induced by sending pulses at a certain particular parameter range. Now, what happens is that imagine this pink circle shows the focal spot size, focus. Now, what you observe is that if the parameters are right, there starts to form these highly regular patterns in a self-organized fashion. So, in other words, we are not engraving them one line by line, but our spot size is much larger than 1 unit. But the structuring occurs in a completely self-organized fashion at a scale which is smaller than the focal spot size. Okay. No one has explained how it happens. We are working on it. Still under consideration. Every help is welcome. Anyway, so what we use is, again, multi-stage amplified fiber lasers. This was developed in a collaboration with Ömer İdday of Bilkent University. So we used his fiber lasers. But Ihor Paulo was a postdoctoral researcher in his group back then. So he did actually design the laser, then he repeated the design when he came here. So we are actually using that. So this is actual video of a process. You see here live in real time how the surface of a piece of polished silicon becomes structured within seconds, okay? So, certain parameters of the laser, it's 1030 nanometer, 370 femtosecond, up to 1 watt average power. This is not a very high average power, okay? This is quite a moderate average power, by the way. And it's about what happens to the surface is shown here in scanning electron microscope images. Yet another tool to mention. Now, take a look. This is our spot size. However, the pattern has at least like 7 or 8 maxima within one spot. So these lines organize themselves instantaneously when you send a pulse on the surface, all by themselves. Okay? This is called laser-induced periodic surface structuring. It's a very phenomenological name, just describing what happens, but doesn't tell about the physical background behind it. we think that it's highly nonlinear effect of multiple ingredients. Okay. Now, what is nice about it is if you if you modify the surface pulse by pulse by scanning this pulse around, you can write nicely continuous and quasi-well-ordered regions, okay, with certain spatial period or frequency. Okay, then you increase the power, suddenly this structure becomes like this, like more like microstructure, which we labeled microcolumn. It's like woven fabric, looks like that. Now what we did is, now we have four different surface structures at hand. The flat, untouched silicon. Then you push the power to maximum, you just engrave a line, okay, just like regular laser inscribing. Then you have the microcolumns and you have the lips. So 4 different structures are available. Then we studied combinations of them to see how the cells behave on them. And we studied on different maturity levels. 3 to 6 hours is in their early phase of development. We see several SEM images of cells grown on such surfaces. And we used fluorescence confocal microscope, the fluorescence confocal scanning laser microscope in biology department, using light source filters and cameras, highly sensitive cameras, and also scanning system. And here are a few fluorescence images of the cells which are grown on such substrates. Here you see the microcolumnar substrate here. We have alternating microcolumns and lips. Here you have deep trenches on flat surface, etc. Now, by structuring certain regions, microcolumnar, leaving some untouched regions around in between, we see that cells prefer to adhere to the microcolumns, but not so much on the flat regions. They prefer to stick to the structured regions. Here is a video of the motion of the cells in unpatterned flat silicon. So there are the stationary ones with fully grown neurites, there are exploratory ones, there is some proliferation, etc. Many different behaviors are observed. Now here is the border between flat region and patterned region. First of all, you see much less number of cells here. They are much more volatile, they move around wildly. These guys stick, they are more dense, but they don't move around as much as they do here. Okay. Then this is on lips. On the lips pattern, they do move more than they do on microcolumns. So they are more mobile, but not as much as they are on the flat. Okay, so something in between. Nevertheless, it's a good compromise because the number of cells on lips pattern is also not too poor, too small. They kind of also stick here. Now these are the trenches. Okay, they are very, very fixed, not moving too much, etc. Now you compare where the lines are. These are where the lines are. If you boost the sensitivity of the camera, you can see the actual laser engraved trenches. Then now take a look at the microcolumn alternating lips pattern. What you see here, some cells are moving directionally in a fast fashion. Some others are fixated, they don't move around, they don't explore much. Now if you take a look where they move, then it turns out that they move on the microcolumns and they are aligned in the direction of the microcolumns on lips. So you can do automated cell statistics using some image processing tool like ImageJ or whatever gradient, etc. and then come up with statistics on how, with which velocity they move on polished, on lips, and on microcolumns, etc. What is the average distance, traveled distance of cells on each of the different kinds of topographies. You can repeat everything when they are fully mature. Okay, I'm not going to go into that detail. We show you nice fluorescence images, flat against microcolumn. Now, in the late stages, you see that cells also stick to flat. So it turns out that it's not correct that cells don't adhere to flat. It's a long, it takes a long while until they agree to stick. Okay? Long onset time, let's say. Then the structured ones. And they show that they prefer to stick to columns more than on lips, etc. Then you can see that you can line them up. Okay, we are not as far as constructing a neural network, but we can line them up at least to a certain extent, which correspond to trenches. You can run the statistics again using the fluorescence, come up with statistically significant differences between how they prefer to stick to which sort of substrate, and take a look at them. Then you can study their morphologies when they are on flat versus structured surfaces. You can see that on flat, they try to extend themselves, flatten, try to hold to the flat surface, whereas they are more, according to me, less stress, more comfy on structured surfaces. They don't work so hard on holding the surface. And so you can come up with conclusions even after this very simplistic kind of approach. So you see that modification of the surface could be useful in modifying their behavior, in causing them to align certain directions. They have certain preference of adhesion to certain textures, and they don't look very healthy on extremely structured surfaces too, neither on very flat nor on very rough. They like more intermittent region. Okay, and here goes my acknowledgements. I come to the end of my 4 different example studies, in which I mentioned, 6 or 7 different physical tools in the study of biological specimens from molecular level to cellular level. Sarah just graduated, went to University of Mainz. Şeyda is continuing that work. She's around. Çağdaş Hoca is our collaborator. Igor Hoca and Ariane are our collaborators. We also got quite a lot of help from Prat and the rest of the team. And I thank you for your attention.
Thank you for the nice speech. Having these topics, biophysical concepts discussed and covered here by you, I think it is a great chance for us, because these kind of topics are even studied rarely in the world, and now discussing biophysical concepts here. Thank you, really. And this was the seminar session, but it doesn't end here. We have now a tea session, so please join us outside for tea. And if you have time,
if the audience has questions.
Oh, of course. If you have questions, you can ask, of course. But don't escape anywhere after the questions. And if you have time,
10 more minutes, yes.
Maybe you can join us for the tea. Thank you.
Thank you. No questions? Okay. Should you have questions, don't hesitate to ask me whenever you like. You can come and visit our group. You're welcome to visit our group. our group has, of course, evolved. This is not one of the most recent photographs. It's before COVID. Now we have other people. Some of them went away, graduated. But you're welcome whenever you like. You can contact me over email, but I tend to give very late response if I do at all. So that's because of my schedule, very tight schedule, can't keep up with everything. But if you are very highly interested, be insistent, okay? Insist, okay? So if I see a mail twice or 3 times, that it makes a difference than seeing it once. So don't worry. It's just my it doesn't signify that I'm not interested. I'm just not coping with everything within the time. Okay, that's all. So, but I can point you to other members of the group who you can go and chat about all these topics. Şeyda is here. She can also answer this last part that I showed. Şeyda is doing more work on that. So, yeah, I guess that's it. There are more stuff we do in our lab. If you're interested, we can talk in another at another time about those topics later. Okay. Right then. Thanks for having me.